therapeutics has changed significantly in the last just 13 months. That is amazing. I mean, usually you don't see that much progress in 13 months. I had a huge advantage. I kind of cheated because I came into the scene of the AHCF Medical Advisory Board in 2012, like Kate mentioned, right after the gene ATP1A3 was linked with AHC. So I kind of missed all that painful, growing pains of not knowing what the main most common gene was, and got right into it when it had already been discovered. So little advantage. All right, we're going to take this back to basics. A lot of you may already know some of this information, especially if you've spent a lot of time in doctor's offices or you have a medical background, but we'll make sure everybody gets up to speed. So obviously the body is made up of cells. Here's our different types of cells. They all have the same DNA in them, which is really interesting, even though they look very different, except for the red blood cells. The poor red blood cells lose their DNA. They start out life with DNA and then lose it, which means when you do genetic testing, you're probably using either you're using the white blood cells for the most part, occasionally other tissues, but mostly the DNA that's inside white blood cells. If you go inside a cell, there's the cell there. I don't think you can see my cursor. We have the nucleus, or the heart of the cell, with the chromosomes, 23 pairs of chromosomes, like the infamous testing company that used that in part of their name. The chromosomes are kind of fuzzy because the genes are kind of coiled up within the chromosome. And if we uncoil that, we see this double helix of life, this DNA strand. And each DNA strand is, this is what the genes are made of. They're made of DNA. So DNA is the substance and genes are the recipes. In a given chromosome, there's probably about 1,000 gene recipes inside that chromosome. And again, our chromosomes comes in pairs, one from mom and one from dad. Here are some chromosomes. You may notice something unusual about these chromosomes. These are not human chromosomes. These are gummy chromosomes. This is an artist rendering of chromosomes, but I love this. They're beautiful. The chromosomes that we would see on a karyotype or chromosome photograph are not quite as beautiful, not quite as tasty. We don't really need to talk too much about chromosomes to talk about the genetics of AHC, but I will point out that ATP1A3, which is, again, the most common gene and not the only gene. I don't mean to exclude those whose loved one maybe has a different gene behind their AHC, but ATP1A3 is here on chromosome 19. The main point of that would be that it's not the X or Y sex chromosomes. So there really are not differences in inheritance pattern, or to my knowledge, even expression of the condition in males versus females, because this is a regular chromosome not linked with sex. So inside your chromosome, sorry, I got rid of, there we go. Sorry, I'm having a little technical issue. There we go. Inside your chromosome, you have your genes, and then the genes make protein products. In clinic, I would say that the chromosome is the cookbook. The gene is maybe the recipe to make an apple pie, and then the protein product is the apple pie itself. So we have a gene instruction that tells us to make something, and that's what we really need to make sure we make correctly. And if we zoom in on this, it's actually a little more complicated. It goes from not just from gene to protein, but there's an intermediate step. So you start off with your DNA message, and this is made up of made up of letters, which are actually called nucleotides. They're letters that stand for chemicals, the 4 chemicals of life, but I often just call them letters. The DNA cannot be directly made into the protein product. They go through an intermediate step called the RNA step. So that's the little messenger. That's actually what the M is for, mRNA or messenger RNA. And it's actually the RNA that is translated by the body into the protein. And you'll see these are grouped into sets of 3. So every 3 letters in the genetic code makes one piece of a protein or amino acid, which I like to call the beads, like the beads on a string. So that's how they start off their lives. They start off kind of in order there in a nice line, one bead after another. But most of our proteins are not straight lines. Then we would be a straight line. And we're obviously very 3-dimensional. So the proteins are then going to curl up into some kind of a shape, a different shape for every protein. And what holds the shape is some chemical bonds in between the amino acids that cause them to, like hairspray, they kind of hold their shape. So there is a amino acid or bead model, obviously a necklace or a bracelet. And there is actually the ATP1A3 protein product there on the right. So if you run into it on the street, there it is. That is the ATP1A3 protein product, which is a subunit of a pump. And we'll talk about that in a bit. All right, but you're here to hear about gene changes. So let's talk about some gene changes. These go by a bunch of different names. So the old term that I still slip up and use too much of the time is mutation. The term mutation has fallen out of favor for a couple of reasons. One is that the connotations. It sounds a little bit like mutant, which is not a very nice word. It really comes from Latin to change, so it was never really meant to have a negative connotation. The scientific problem with mutation is that not all mutations are harmful mutations. Any change in the DNA is technically a mutation, even common ones, even ones that 59% of the population have. So we don't like that term so much, but it's a convenient term. The correct term in 2026, or the preferred term, is pathogenic variant. Variant meaning difference from the typical spelling of a gene. Pathogenic meaning disease causing. Also rather cold and clinical and hard to say. So I like to go with gene change. Now that's after we define that the gene change in question is the cause of somebody's condition. Usually that's what we're talking about, those kinds of gene changes. So here's an example of a gene change. So in the DNA sequence, the letter G with the arrow there, the purple G, that would be the typical spelling that most human beings would have. That probably goes with having good health in the absence of symptoms. But this person has a T instead of a G, and that will cause in the mRNA, a different letter to occur. I'm not totally sure that's right. Oh, sorry, that's right. And the mRNA uses a little different language, so it becomes a U. This is correct. I didn't make this figure. But the problem here is that the protein should have been a W, which is a tryptophan. And here it becomes an L or a lysine. So by changing UGG to UUG, you end up with a different protein amino acid inserted into the chain of beads, and that can lead to some problems if the body can't tolerate the new amino acid. And sometimes the body can, but oftentimes it cannot. So if you change the beads, you're going to change the shape of the protein and maybe the function of the protein, which is the issue. Here are some examples of gene changes, and the little abbreviation here is just showing the amino acid change. So on this first one, at position 129 or bead number 129, what would have been a valine becomes a methionine. So there's a 3-letter code or a 1-letter code to tell us which amino acids we're talking about. And in this diagram, they're trying to show you that different conditions are caused by different changes, although some amino acid changes are capable of causing more than one problem. And some health problems like AHC can be caused by different changes. So it's not a one-to-one system. You're not meant to really memorize this. I just wanted to show the diversity of AHC as not the only ATP1A3 related condition, and that there are many different gene changes that can lead to AHC, which we'll get into a little more. All right, can everyone still hear me okay? Because now are we still good?
Yep. Yes.
Okay, good. Sorry, having issue. I'm not seeing the same thing I was seeing before. Sorry about that. All right, patterns of inheritance. So it's important to understand how genetic conditions like AHC can occur and how they don't occur, because this can affect your family, your other loved ones, maybe the siblings of your child who's affected with AHC, but potentially your child with AHC themselves. So this is an autosomal dominant condition. I think every single gene that's been linked with AHC, or even a lot of the mimics of AHC, are autosomal dominant. So I call these bicycle genes in my clinic. A bicycle gene means that you've got 2 wheels in your bicycle, and as long as both wheels are functioning properly, you're going to be good riding your bicycle. But if one wheel is not functioning, it's not a unicycle, it's a bicycle. It needs 2 wheels. So one non-working wheel or one non-working gene change is enough to cause some trouble here. The term heterozygous refers to a person with one working and one non-working copy. And so in this picture, the poor dad here, he is heterozygous. He is shaded in blue. And this couple has had 4 biological children. And an average of 50% of them have inherited dad's gene change. And the reason is dad's got 2 copies of this gene, one affected, one unaffected, where the arrow is pointing. And when he goes to have children naturally, and there are ways to have children less naturally, but every option is right for different families. Those who choose natural conception will have a 50-50 chance of which copy is passed to each child. And here it does not matter what the gender of the children are. So you see that it's half his boys and half his girls are affected. It doesn't work out this neatly in most real life families. You can have all your children unluckily affected. You could be fortunate and none of your kids inherit a heterozygous condition. And this is just a giant family. When you take it out and you zoom out generations, showing that the 50-50 pattern continues through the generations. But this is not what we typically see in AHC. It's usually something called a de novo dominant gene change. And that means if parents are tested, and nowadays a lot of parents are tested at the same time as the child, when they have whole exome or whole genome sequencing, they do what's called a trio if both parents are available. And they'll test mom and dad and child. and they'll tell you that the child is affected and the parents are not. And I had a patient in clinic today, a parent in clinic today, very alarmed by that. That wasn't AHC, but it was a different neurogenetic condition. And they said, but wait, we're sure we're the parents, and we're both the parents. I said, yes, you absolutely are the parents. In fact, the lab can tell me when I submit a sample from a person who's not biologically related to the main patient. So you are the parents, but you don't share the gene change. So this can be a perplexing concept for a lot of folks because we think genetic equals inherited. But here, mom and dad, they don't have the gene change, only this one child. And it is just the one child, it's not their three siblings. So how does that happen? And that happens because a change happens either in the egg or sperm cell that forms the child, or possibly a little bit later on when the child is an embryo or a zygote, which is like a very early embryo. It has to be extremely early, otherwise you won't see it evenly distributed in every cell of the body of the child. We think most of the time it happens in the sperm cell, not to blame the dads, but sperm cells are a little more vulnerable to these new changes compared to egg cells. And so how does that happen? Do you have to live near a radioactive dump or something to have your cells, your DNA change from generation to generation? The answer is absolutely not. All of us have these de novo or new gene changes in our DNA. I have them, you have them. In fact, we probably have about 44 to 82 changes if you were to check all of our DNA against our biological parents that are different. And this does not require any risk factors whatsoever. It's pure luck where this gene change will happen. And if it happens in an important gene, then you can, of course, get a condition from it. And so basically, to make new cells, to make sperm and egg cells, you've got to make new DNA to put into those cells. So the DNA will unzip itself into two halves, and then it will regenerate the missing half until you've got two separate pieces of DNA. And it's remarkable how well this does work, but there is an error rate of this copying, just like if you were trying to, copy a list or something with a pencil onto another piece of paper, there may be an error. And this happens sometimes in very important places. And it's nobody's fault. This is really the important thing to know. So what genes are we talking about here? So I keep mentioning ATP1A3. It's about 75 to 80% of AHC, which is going to leave some of you without this diagnosis. The OG gene, the original gene that before 2012 we thought was the culprit, was ATP1A2. That can sometimes be seen in people who do meet the clinical criteria for a diagnosis of AHC, which we won't talk about the criteria in detail today. This is probably now better known as a familial hemiplegic migraine gene. So it causes migraine headaches that cause paralysis on one side of the body, which does overlap a little bit with what happens in kiddos with AHC, and it's also kind of helped by sleep and things like that. But that gene is less commonly seen now in kids who have the full symptoms of AHC. Down at the bottom are some occasional AHC genes. These are actually going to be called like AHC type 2 or type 5 or something like that. So they do count as AHC, but they are less common. And these are, again, not really in the scope of my talk today, but very important for those who have them. But some people go away with no diagnosis, no genetic diagnosis, just a clinical diagnosis. And that's very frustrating most of the time. And why is that? Why are we not perfect at finding this? So one reason may be that there's probably some other genes out there that we have not discovered. I think that's probably true for most rare, fairly severe pediatric conditions where we just can't find a diagnosis for a child, is we haven't found your gene yet. And if you use a lab that offers continuous reanalysis or gives you a free one-time reanalysis later, or you're in a research program, you maybe one day get an update that they found your child's gene, which would be a good, exciting development for most people to have that answer. Another theory, which I don't think is at play here in AHC, is that could it be not a gene? Meaning, could it be many little weaker risk factors combining together? So things like learning disabilities, even some ADHD, some of these kinds of conditions we don't think are due to one gene. There's even epilepsies, childhood absence epilepsy, the very common staring spells, not caused by one gene, probably caused by multiple genetic risk factors. But the rule of thumb is that the more severe the condition and the more the child stands out in the family tree as having a unique condition, it's probably not a complicated inheritance. It's probably a gene that we just haven't been able to find for you. And then finally, some people probably don't have AHC. They probably have something else that would probably be better served with a different label. So here's some examples. There's a pyruvate dehydrogenase complex deficiency, where kids cannot use sugar for energy, can have some similar movement disorders. We have mitochondrial conditions, which are often caused by changes in the DNA that does not live inside the chromosome. but lives inside the batteries of our cells. Various neurotransmitter disorders, which Kathy Swoboda, who's involved with AHC, also studies. And then finally, just a whole bunch of other movement disorder genes, CACNA1A, SCN1A. So these are probably better served, these individuals who have these gene changes, probably better served with going with that gene and what that gene does and not being looped in with AHC in most cases. All right, so focusing back on ATP1A3, and I'm watching my clock here to make sure we have enough time for questions. This is an diagram that one of our PhD students made. Is beautiful, and it's showing the product there. There is not the product of the gene, but the pump in its entirety. So there's more than one subunit or piece that makes up the pump that we need to do this function here. ATP1A3 makes up a piece of the pump, and there's other genes that make up other pieces of the pump. So it's like it has to be assembled together into a final pump. And AHC is considered a fancy medical word, which is a made-up medical word, a pumpopathy, apathy meaning condition and pump meaning pump, because this is not a channel. A channel is where these little charged molecules called ions, if you remember chemistry class, if you took chemistry, a channel allows ions to kind of flow just kind of passively and lazily through the channel. But a pump requires energy. So we take ATP, which is the energy that we make in our bodies, and we actively pump like if you're pumping water out of the dike back into the ocean. Here we're pumping some sodium ions outside of our cells. These are the brain cells that we really do this in. And we're pumping our potassium ions. The sodium is the Na, that stands for sodium. Potassium is K. I don't remember why potassium is K, but K is going to be pumped inside the cell. And what you would like to have is sodium outside the cell, potassium inside the cell, and that sets up the right kind of electricity ratio on the outside to the inside of the cell. So we call that the electrochemical gradient. And if you don't have your pump functioning properly, you won't quite have the electrochemical gradient that you want, which makes it harder to fire signals from cell to cell, because that's what the gradient kind of does. And on the right and left is showing the loss of the sodium back into the cell passively and the loss of the potassium out of the cell passively. That's what we're fighting against by pumping things back into their proper places. So ATP1A3, again, makes up part of that pump. It's probably 75 to 80%. It really depends on who you read. And most kiddos and adults who have a change in this gene will have one of these three changes, D801N, E815K, or G947R. Some people have rare gene changes that are not one of the top three, and that's fine too. But these are the most common. And since they're also the most common, they're the best studied. We know there's some trends. So when we talk about genotype-phenotype correlation, it's a fancy way of saying, can we predict the symptoms from the exact spelling change in this gene? So sometimes we look at the genotype, like this one, D801N, and we try to make some predictions about which AAC symptoms that person might experience. So if we look at E815K, that one tends to be a bit earlier symptoms, more seizures, more motor and cognitive impairment, and a little more rapid progression. Now, this is a generalization. I always say your mileage may vary. So your child may have E815K and not fall into these categories, but if you look across the population level at all the children and adults with this, these are the trends that we observe. If you look at D801N, this is the one with a little bit more heart risk. So short QT interval, which is measuring part of the EKG, and this can be a setup for heart rhythm. So I believe that most of us, most children who have, and adults who have been diagnosed with AHC, especially those with D801N, have probably been advised to go get an EKG or go see a cardiologist to check this out for them. It doesn't mean that everyone experiences this complication. This is just something to watch for to try to head off any problems. All right, and now conversely, this is not the focus of tonight's talk, but not all ATP1A3 is AHC. So occasionally we have people misdiagnosed with AHC because their genetic test report said they had ATP1A3. So it's part of a huge family of what we call ATP1A3 related disorders. Say that 5 times fast. AHC is probably the second one described. RDP, rapid onset dystonia Parkinsonism, was linked with this gene back in 2004. So I think it took about 8 years for them to figure out that AHC was also this gene. There tend to be different gene changes, not the exact same gene change, just the same genetic recipe. And then there's a bunch of exotic ones down here that what we also see. There's probably about 12 now, which is pretty amazing because I did not see that coming. And right, you've seen this before. This just shows these abbreviations are the different conditions, and they tend to go along with certain gene changes. a little bit more, more specifically, like this one here, RICA, which is the one I study in my clinical research. Most people have arginine 756 cysteine. That's, if you see that mutation, you're probably dealing with RICA. So you've got to be careful when you're reading a test report, which we'll go on to now, that you interpret it, and you as your geneticist, hopefully, or your neurologist, whichever provider is guiding you, not really the parent's responsibility, although I know parents take a very active role in researching these things and helping their providers. So genetic testing comes in different sizes. I just noticed that the person I stole this from misspelled coffee. Maybe they hadn't had their coffee yet there. But small, medium, large, ginormous. And most of our kids are probably getting large and ginormous tests. It is a rare day when someone says, I think this is ATP1A3. Let's just test that gene. I've done that once in my life. I've zoomed right in on it, but usually we're not that specific. So you could just test that gene if your doctor was really, really on it, or your provider. Medium would be like a collection of genes. So we're fortunate now that ATP1A3 is on different seizure panels and things like that, so that even if a provider does not recognize AHC, in the old days you had to recognize it. Now you say, well, this kid has seizures. Let's send a seizure panel, which is 320 genes related to seizures, or movement disorder panel, or developmental delay panel, depending on what symptom has caught the attention of the family and the provider. And then if this gene is listed on the list of genes that gets tested, then you get to detect your gene. So you don't need to be smart enough to know what you're doing, basically. You could be even less smart if you go with whole exome or whole genome sequencing. This, though, is a test that does require the provider to be diligent and feed in information about the patient to the lab, which will then look at all the genes in the body. So we have about 20,000 genes crammed into those chromosomes, and they will look at all of them and return results that they think are relevant to the patient's situation. They try not to go crazy off topic with random stuff. It's really about, okay, this child has seizures, delay, and maybe hemiplegia. Let's look at genes that could explain that and see if there's something that we can detect in one of them. We've had this test now since 2011, and I really do think it's probably helped us increase the diagnosis of kids with AHC and adults, and helped us to not be quite as rare as we used to think this was. The problems with exome genome, it is a really complicated test to order. It's not really that user-friendly. You do want to set it up with parents or relatives if you can, if you have that luxury of having those biological relatives available. We don't always have that luxury, and we can deal with it if we don't have that luxury. The overall rate for a child presenting with global developmental delay or intellectual disability with the exome genome test is about 50%. So about half the time we'll find the diagnosis, and sometimes it's AHC. And it also comes with an option to find some off-topic results, which we call the medically actionable incidental findings. So this is a test that's probably mostly employed in the genetics clinic with a genetic counselor or a geneticist, because it is a little bit of a production to do, but it can be very rewarding. The exome was the older test since 2011, and now post-pandemic, a lot of us, and just for me, just the last year or so, transitioned to mostly using the genome. The yield is probably only about 10% better. It's not a miracle better, but it does find some stuff that exome can't find. ATP1A3, though, really should be able to be found on the exome, the smaller test. All right, when you get your report back, though, this is another big kind of pitfall. If it's positive, it's positive. And usually if it's positive, it stays positive. It's a rare day, although this happened to me this week, where a lab writes to me and says, oh, that positive result from insert year here, oh, we changed our mind. It's a no, or it's a maybe. Most positives are there because of strong evidence that this variant can cause this condition, and that's not likely to change. Negative means we didn't find anything. It does not mean your child does not have a genetic condition. It just means the test was not successful in finding a genetic condition. I have patients in my practice that I'm confident have a genetic condition for many reasons, can't find their gene change in 2026. And then finally, we've got the infamous variant of uncertain significance, or VUS, if you like to pronounce it that way. This means there's a difference, but the lab doesn't know if it's a bad or harmful difference. Maybe it's a helpful difference. It could be a neutral difference. It just means it's not the typical spelling of the gene. Extreme caution with these, because sometimes they are the real deal. They are the cause. But if the child's symptoms don't fit anywhere near AHC and they have an ATP1A3 VUS, maybe they don't have AHC. So you really want to be skeptical and careful. Just use caution with those. So this, again, has allowed us to find kids and adults that we never would have thought about this before, maybe because their symptoms are atypical, or maybe because the physician or the provider just does not have enough experience to know what they're looking at. And this allows us to realize that the range of symptoms is broader than we thought, because if you only test the people you think fit the textbook description, you're going to miss all the people who aren't quite typical and think that they don't really have AHC. So we're kind of broadening our horizons of what this can look like. And here's a test report. This is an ancient test report. Unfortunately, not an AHC test report because I couldn't find one, but this is for some kind of a RASopathy condition. Well, here's the gene called RAF1 or RIF1. And what you're looking at here is just your child's test report will have the name of the gene that's being assessed here, the change in the letters of the genetic code right here. So this is letter number 1423. Usually it would be a T, but on this copy of the gene, it's a C. Here it's showing you that this changes the amino acid from phenylalanine to leucine at number 475. For any math people, if you divided 1423 by 3, you should get 475 because it takes 3 letters to make up a bead or an amino acid. Heterozygous, there's that word again. This is a dominant condition. So only one of the 2 copies is going to be affected. Homozygous would be both copies. We don't see that in ATP1A3 or in AHC. It's disease causing. We would now call this pathogenic variant. Again, this is old. And this is telling you that you can be confident about this. And then down here, the lab's going to give you some information about why they think it's pathogenic. Maybe they've seen it before, or maybe they've done some modeling on a computer that shows that they think it's going to affect the gene. And here you got some guidelines. So I'll make sure we get to the next part. This is the most important part, I think, of all. This is where are we headed with this? So sometimes we can't find your gene, and that's, I like to tell people that we're just pausing the genetic workup until we get smarter. We're not done. We're going to continue to look for that gene. But once we have the gene, this potentially opens up some new options here. So if you don't have a gene, you can still have a clinical diagnosis, the gene being a genetic diagnosis. It's still useful to diagnose AHC without a genetic cause, because that way we can understand a little bit better what to expect in the future based on other people's experience with AHC. And we can treat the symptoms maybe with agents that have been shown to be helpful. These are not genetic therapies. These are just drugs and medicines that seem to treat symptoms that are seen in AHC. But if you can get the genetic diagnosis, you might even be able to predict the outcome better, like we talked about with the heart rhythm versus epilepsy and the 2 most common gene changes. You can then even think about trying to change or fix the gene, and that in order to change or fix the gene, you have to know what gene change you're dealing with. So here's 3 categories of genetic therapies. One would be where it's a medication. It's not really a gene changing therapy, but you are going to not just use any old medication. You're going to address the underlying issue, which is that sodium potassium pump that we talked about. You're going to try to make the pump work better. So that's considered a symptomatic therapy. You stop taking it, you stop getting the benefit. Down the bottom, there's disease modifying. So there's temporary genetic therapies. There's supposed to be a Band-Aid on the left if you can't tell. The genetic Band-Aid, it temporarily improves the way the gene's working, but it's not permanent. And on the bottom, you've got your permanent one and done fixes, or we hope one and done. We hope they last the life of the person. So here's an example of the genetic patch in the middle category. So just showing DNA, RNA, protein. And what can you do? You can stick on a tiny little piece of RNA. It's called an antisense oligonucleotide, or ASO, because nobody wants to say that more than once. The ASO sticks on right here. And it's actually, in this case, on the left, what it's doing is it's making the RNA convert into a different protein, a green one instead of the purple one that it would normally make without alteration. On the right, you've managed to stick on a piece of RNA that blocks the RNA and says to the body, hey, don't use that RNA, we're blocking it. No protein comes out. Remember, you've got another copy of this gene that's presumably healthy. So the person on the right now is going to work off their healthy copy and ignore their copy that has a change in it. So the advantage here is you can undo it. It's going to wear off. You usually have to dose these things every few months or so. If you don't like the effects, great, it wears off. If you do like the effects, it's not good, it wore off. You have to give it again. You can put pretty big genes. You don't have to fit genes into a package to get them into the body. So you can modify large genes. You don't need to worry about allergies to the thing that you're putting the gene in to get it into the body. And it's really inexpensive. So this is an area that is being explored by the folks at Northwestern, and I have it coming up where they're doing their research now. So this is now at the, I believe, mouse phase. Oh, at Jackson Lab. They're doing this in mice. It can be a little hard to get these into the nervous system, usually given by a spinal tap. We are seeing similar, they're not technically ASOs, they're called small molecule RNA splicing modulators. These aren't for AHC yet, but they're being developed for things like Huntington's and spinal muscular atrophy. And it's a pill that you take every day. So the kiddos with SMA are already, and some adults are already taking these daily pills instead of a spinal tap to get something like an ASO gene patch into their body and help the functioning. All right, so we covered this already. Now, is it okay to knock out one copy and have the other copy? We think the answer is yes. We think that most people would be better off having just one working copy than a copy that is doing something we don't want it to do. So this may be a really smart approach. And here, this is the slide I was talking about. So they did it in cells first to make sure it works. And now they're trying it out in mice. It's at Boston Children's in collaboration with the Jackson Lab, which I think is the source of the mice. So we're not up to humans yet. They also are working out how to attack the correct copy. So meaning the non-working copy of the gene. You want to turn off the correct copy. You don't want to turn off the healthy copy. And they came up with a couple systems. I'm not going to discuss that today because it's we're running short on time, but there is going to be a way to target it to turn off the copy that you would like to turn off. So that's one really promising area that's getting a lot of attention. The other one is gene therapy. So the original gene therapy is just, let's stick a new copy of the gene into the body. And we put it inside a little package called a vector and a vesicle, and it goes inside the body and goes into the central nervous system, goes into your DNA, and voila, you have a working copy of the gene. This did not work that great in the mice. Some of the mice did well, some of the mice did not do well. A little hard to get it into the brain, that can be a challenge. And then there's the issue of not removing the non-working copy. So it was still there, potentially causing some problems. So this was probably not the way to go, the adding a copy, because I don't think people I know, people with AHC do not typically have a missing copy. They have a copy that's doing something they don't want it to do. The Dr. George from Northwestern has told me we should not use the term dominant negative, but that is the typical term used in these kinds of situations. We're just going to call it a dominant effect. But basically, if you have a copy, a subunit of the pump that is not the correct shape because the non-working gene has made it not work properly, it's going to combine with all those other pieces that make up the pump and maybe crowd out the healthy pieces. So now you've got the not healthy pieces combining with the other subunits that come from other genes to make a pump that's not working. So if you can get rid of the copy that is not desirable, that could be a better solution than simply adding more pieces. That's like this Lego tower. If you don't want the orange ones, just putting on more Legos is not going to help you. You want to get rid of the orange Legos. Sorry, orange Legos. I love orange Legos. All right, so this is the press release coming. Well, sorry, the next slide is the press release. But last week, someone sent me a New York Times article that maybe some of you have read about the Frost family and some of their funding efforts towards a new initiative. And this is super exciting. I'm really, really pumped about this, no pun intended. So base and prime editing. Base editing is not really what's being used in AHC, but that's just going in there and actually altering the gene in the human being. In this case, we're up to mice, not human beings. But can we just change the letter in the person permanently? That would be maybe the ideal solution. Prime editing allows us to do more than just change a letter, but it can also change the letter. It's just a little bit fancier approach. And right now in 2025, this is what this article is on the left, which are a lot of names that you may be familiar with on the left there. They were able to change about 74% of the cells from the mutated copy or the changed copy into the typical sequence of letters that you would want to see. And then when they gave it into mice, they actually helped their pumps work better. The mouse behaved better, meaning they did not have hemiplegic spells. I don't mean that they were, less rowdy mice, but they were able to have better neurological functioning, and then they survived better. So that is super exciting. And again, this is mice. But now we're really poised to start to expand this to humans. So the Center for Therapeutic Genetics, this was the press release or the New York Times article just last week. I had to make these slides again to make sure we got this in here. A collaboration between the Broad Institute, which is MIT and Harvard, the Jackson Lab, and Boston Children's, and trying to come up with a way to provide base editing broadly for many, many rare conditions so that we don't have to reinvent the wheel every single time for every single rare condition, every single child. So this is really exciting, and this is focused on base and prime editing, and this is hopefully coming soon. There are no clinical trials right now. If you go to clinicaltrials.gov database, you will just see some databases and registries and things. You will not see any opportunities to enroll in a clinical trial. but I am very excited to see the progress, and I'm hoping that this is coming pretty soon. And once again, the generous contributions of families have your funding makes a difference. So it's unfortunate that parents who are so busy taking care of their children have to do golf outings and 5Ks is what I'm quoting the article here. But those funding efforts have really paid off. So you're driving this, which is amazing with the help of the scientists. And I want to thank Dr. George, who continues to coach me about gene therapy and correct my misconceptions, because I am a clinician, not a scientist, just kind of a fan of gene therapy. All right, thank you very much.
Oh, thank you so much, Dr. DeBrosse. Obviously, a very informative session. We knew it would be. I'm sure that there are questions. I am going to turn it over to Kelly Androska, who is here. I'm sure most of the community is familiar with Kelly. She's a huge stalwart in our family committee, family support team. So Kelly, if you want to take over the Q&A session, that would be great.
Sure. The first question is actually mine. So my daughter's initial genetic testing did not show a pathogenic variant of ATP1A3.
Okay.
However, a year later, they looked further down the chain and they found ATP1A3 5801N. So my question is, what does that mean further down the chain? And then does that point to maybe the severity or the severity of the disorder or being more mild?
Yeah, so I think I'll have the second one. So I think there are that fancy word, that genotype phenotype correlation, that D801N tends to have a little more cardiac risk, which again is a minority of our kids. We just focus on it because it's scary, right? And maybe a little bit more mild. I think ultimately trends are most helpful when your child is first diagnosed and you're trying to, maybe you have a newborn or a small child and you're wondering what happened to other families. As your child gets older, they've kind of shown us what they're doing, and maybe trends are a little less critical. Like reading about other kids is less critical than knowing your own child. But I think there are some general trends. But again, it's one of those, your mileage may vary. Did you find that she did seem milder or in any way compared to other people that you've met? Or
she seems more mild to me.
yeah.
Yeah. I mean,
Yeah.
compared, yeah.
Yeah. Good. And I think I know her, but I'm trying to think about which way she may be milder. And none of the kiddos right now who are, just full disclaimer, who are in the AHCF are my current patients. I've seen a few in the past, but I mostly come to the conferences to see everybody at the conference, which is great. And I have a couple of patients with this FIPWE-RICA variant, which isn't really AHC. To answer your other question, I wonder if the technology just improved. So I don't know what the first test and what the second test was. I have a woman in my practice, again, she has RICA FIPWE, but I sent her testing in 2017 to a very prestigious research lab. I won't mention the prestigious research lab, but it was very prestigious. We did that because it was free. And because at the time, it really wasn't an easy gene to get tested back in 2017, as easy as it is now. It came back negative. Then she had a child because, again, this was a little milder variant of ATP1A3 disease. And then last year she came back to my clinic and I said, well, we got a free test for a whole bunch of different genes. Do you want it? Do you want to get all these genes checked? We'll find the real culprit. Well, not only was it ATP1A3, but it was the exact mutation I thought she had, that we missed it in 2017, which was super embarrassing because now she has a kid that could have inherited it. He did not, thank goodness. But no, to this day, I have no explanation why the technology failed, but the technology does get better and better. So although you get what's called a free reanalysis when you do an exome or genome that doesn't work, what that means is that the lab is going to go through the data. They don't save the DNA, they save the data. And then they'll go through the data several years later, whenever the family decides to do it, in light of the new symptoms of the child and the new gene discoveries. But the basic data is the quality that the data is. It's like, the resolution of a photograph or something, right? The resolution's not going to get any better. It's still the 2017 technology or whatever, whenever the test was originally run. So a lot of people advocate, if you don't have a diagnosis and you really think it's a gene that you missed somehow, not a different gene, maybe consider running the test from scratch, maybe a different laboratory, a different methodology. Working with a geneticist can help you identify whether there was something, I hate to use the word substandard, but something not great about the original approach. But I think it's just the technology gets better. That mutation seems straightforward, and I don't know why they would have not found it, but I'm not an expert on the intricacies of that one, if that makes sense. But
yeah.
Long-winded
Okay,
explanation, sorry.
that's
Like
okay.
it was fine for others. Hopefully that was helpful for some
Um,
others too.
yeah. If anybody has any other questions, please put them in the chat.
Oh, I could have gone
Um,
for another 11
and
minutes on that topic. Sorry.
yeah, in the meantime, we have gathered a few offline here, so
Okay,
I will start reading those.
please.
So if two children have the same ATP1A3 variant, why can they look so different clinically?
Because nobody is one gene, right? We are 22,000 genes plus our life experiences. So there's probably a lot of randomness in life, as well as the influences of all the other genes. So we don't exist in a vacuum. There's probably other genetic factors that modify, as well as any effects of things that happen to our bodies. Whatever it is, it's not, I don't mean to imply that it's a parenting decision or a toxic poisoning or anything like that, but the accumulation of potentially things like spells and probably more so just the other genes in the body may be driving these differences. So that's why genotype phenotype correlations are rarely an absolute lock on how someone's going to turn out with a given symptom. If you think about sickle cell anemia, every single child with sickle cell anemia, the classic form, has the same exact mutation. Some of them are doing amazing and some are having strokes. And it's not one gene. And everybody with achondroplasia dwarfism, almost all the patients have the same mutation, I think 96% or so. And there's a range of outcomes with achondroplasia dwarfism. So I think it's just all the other things about our bodies and our DNA. That's a great question.
Okay. Here's one that just came into the chat. I can't find any other cases of my daughter and my variant. Is that why it's considered a VUS?
Probably. So the things that will get it out of the VUS category, number one is having other published cases where for different reasons people are certain. So if there's 43 other people published with that variant who clearly have AHC, then we're going to call it pathogenic. If you were able to do an animal study with it, like put it into a mouse, this is usually prohibitively expensive, and see if the mouse gets AHC, then sometimes those animal studies can prove that something's pathogenic. What we don't usually have the luxury of in AHC is multi-generational, what's called segregation studies. I think the fact that I didn't catch that there was a mom or a dad. If this person is healthy and shares it with their child, it's going to make the lab think twice a little bit about calling it causative. And the reason is that most AHC, not all, most is de novo. It's inherited from two parents who don't have it. And the fact that it's only the child with the symptoms who has the gene change helps the lab to think that it's pathogenic. If a parent has it and they're healthy, it could be that variability, right? That the mom and the daughter are not, or the dad and the daughter are not clones of each other. So outcomes can vary from generation to generation. But then the lab's wondering also, maybe it's not the culprit, and that's why a healthy mom shares it. So getting more cases out there, eventually this will happen. So eventually whatever lab you used and all the other labs, they share their data. Anytime you do a genetic test, you sign a waiver that says, usually, whether or not you know you're signing it, that your lab may share de-identified data with other scientists and labs. That means they're taking off your name, they're taking off your birth date, but they're sharing the raw data so that we can get smarter about this. And someday there may be either a whole bunch of people who seem like they have AHC who have that variant, and the lab's going to say, bingo, it's a positive, and you're going to get a reclassification. Some labs you have to ask for that reclassification, like your doctor has to write to them and say, hey, any updates? And other labs will automatically do that and kind of inform your doctor. And then perhaps, though, there'll be a whole swarm of people who maybe they were submitting their DNA because they have a toenail disease. I'm just making that up, just kind of like being light about it. But they don't have AHC, and they all have the same variant, which may imply that it's not related to the nervous system. So I say stay on it. I would never dismiss a VUS, especially if the child has the classic symptoms. You know, the child has classic AHC and a VUS and ATP1A3. I'm darn suspicious that that is the real deal. But if there's something a little atypical about the symptoms and about the variants, I start to question a little bit more. So I say keep an open mind, but just don't get too locked in either way. And again, I don't know your situation, but as a geneticist, I try not to get too locked in, because when you get locked in, everybody closes off their mind from further thought. And that's not usually helpful for patients to have doctors closing off their minds.
And she did just clarify that she is a mom and she is not healthy.
Not healthy. Okay. So that
Not
helps
healthy.
a little bit. I'm sorry to hear you're not healthy, but that helps strengthen the argument if you have similar symptoms to your child. And then we've got two generations with the gene and the gene change. I don't know if it's possible if your parents are living to get them tested. That sometimes not everyone has living parents and not everyone can financially get the grandparents tested. But if we can prove that this is not going back 15 generations, because that would be very unlikely for a pathogenic ATP1A3, but only going back one generation to two people, a mom and a daughter with symptoms, that might help push this into the pathogenic. Or there's another category, likely pathogenic. That's one we probably think it's pathogenic, but we're not 100% sure. We don't have quite enough data. So I don't know if that's an option or not. It often is not for various reasons.
And then a comment came in right after that, that's what happened to her daughter. She was initially VUS with the first panel, but then with the whole genome sequencing, she came back pathogenic.
So that's either because parent data proved it's de novo and then that sealed the deal, or just in the intervening time between round 1 and round 2, they realize that that variant is pathogenic. So sometimes the second lab will just call it differently. They'll see the exact same thing on the machine, like the result is the same, but the interpretation changes because we get smarter in the time it took you from test A to test B. So that can happen as well. So don't lose hope. I mean, I have all these patients who 4 years ago I couldn't diagnose and now I have it. And for some people it's a longer wait than others, but I just tell people we're never done until we've locked it in for you, and we'll just keep working at it with every new tool that we get until we're there. So don't go away from genetics from 20 years is the other lesson. We've had patients who do give up because the tools in the 80s and 90s were terrible. For example, I'm saying that sounds judgmental, but we didn't have very much back then. And then they will show up with their adult child, and we've had the tools to diagnose their child now for maybe a decade, but they didn't realize that genetics had improved so much it was time to come back. So I recommend people come to see me every few years so that they can stay up on what's new, whether your child is diagnosed or whether you're still searching for your gene change or the classification.
And this particular child is she's the only one with that variant. No, to mom's knowledge.
Yeah. And the question is,
side note.
Yeah, for mom is it, and I'm assuming this is not a common variant in any databases. So like in the databases full of random people who've submitted their DNA, we don't see the variant. So it's really rare, just generally rare.
I'll let you know if she comments.
Yeah, because that can be telling too. If it's something that we see in 5% of people, they're going to classify it to harmless because 5% of people don't have harmful ATP1A3 changes. But if it's really rare, even in healthy people, and it's just literally in you and your daughter, I think the chance of it being something goes up, especially if the symptoms fit.
Okay. Thank you, Dr. DeBrosse.
All right, thanks.
Do we have another question?
Yep. Going to the distant future, you mentioned about the difficulty of administering gene editing meds. Will the oral drugs, not the spinal taps, if and when available, have the same challenge as the difficulty of crossing the blood-brain barrier?
Great question. So the oral medications that are being used, again, they're not being developed right now for AHC, but for things like Huntington's, they do manage to get through the blood-brain barrier, which is amazing because that's where they're needed in Huntington's, for example. Huntington's is a degenerative brain disease, mostly of adults. So this drug has actually slowed down the progression of Huntington's by 52% over 2 years by getting through into the blood-brain barrier. And some of our children with SMA, their parents have chosen the daily pill over the spinal tap because it has fairly equal effectiveness. And who wouldn't choose a pill over a spinal tap for their kiddo, right? Gene therapy, actually modifying the gene, I don't think that is coming as a pill anytime soon. But the cool part is that the gene therapy is more often, there are different ones. Some are given as an injection into the central nervous system in some way, but there are some that are given IV. So ideally, an intravenous injection just into a vein. So I'm hoping that at some point we'll be able to do gene editing through a vein. That will just be absolutely amazing as a one and done treatment.
Doctor, thanks for your presentation. My granddaughter was born last month and had eye fluttering shortly after birth. We discovered last week that she has D805G mutation.
Okay.
De novo, likely pathogenic based on the symptoms exhibited, which apparently is previously undocumented. We're in the process of trying to better understand and put together a care team. Based on the unique mutation, would you have any recommendations on where to start? We are based in the New York area.
Based in New York area, you have a lot, you have a lot of options that are not that far. I think, and correct me if I'm wrong, Sharon, do we have a directory of people who are clinicians who are experienced with ATP1A3 and AHC?
Yes, we do. Dr. Sharma's team, a lot of the people out there in Boston and New York.
Yeah, Boston Children's is one of the research areas. Going to a place that's doing research at their institution sometimes does open up opportunities to enroll. You may find out sooner, although if you're connected with AHCF, I imagine you're going to find out pretty quick what's out there as far as clinical trials. Most people just see a geneticist at a major teaching hospital. So we don't expect people who live in states that aren't New York to travel to New York necessarily to see an expert who's doing research. But just I do recommend people, or you're probably already doing this, working with kind of a major teaching hospital with people who see a lot of rare things. My approach to AHC is not that different from my approach to the other 7,000 rare diseases that we deal with, as far as how we connect people with specialists, how we keep in touch with the database of which gene therapy trials are happening, and that sort of thing. So your doctor doesn't have to be an AHC expert, but it helps if they've seen a case or two. You're likely to see that at a larger center. And it's okay to ask the office, the department, when you're scheduling, has Dr. So-and-so seen this before? The secretary is not going to know that off the top of his or her head, but they can ask the doctor and get back to you. I think it is a challenge when your child is a very rare one, because we do think that the research is starting. I read this in the New York Times article. It's going to start with the more common gene changes in order to help as many kids as possible, as fast as possible. But the goal is to get this scaled up so that anyone, even the really ultra rare conditions, can get their child's gene corrected eventually. So again, I think it's hard to say how fast this is moving, but I think it's moving faster than I expected. I was a little shocked to see this article last week and see how far we've come with prime editing. A year ago, this was not on my radar at all. And I talked to Dr. George how to give that talk last year. We talked about ASOs and we talked about the unsuccessful mouse replacement trial. We did not talk about prime editing. So hang in there, get your team of specialists, make sure that you trust your specialists. If you don't, find a new specialist. And you'll know pretty quick if your specialists have your child's best interest in mind and have had some experience with this kind of condition before. Usually neurology and genetics is a good team. And of course, primary care. You don't want to forget primary care. I'm over time here, sorry.
No, it's fine.
That's like me in clinic.
We're okay. We're okay.
Cool.
He said, thanks for your optimistic message and advice. And then just back to mom and daughter from earlier.
Yeah.
They thought mom had MS, but she doesn't. She has some strange symptoms. She has permanent facial palsy that's confusing her doctors. Does that make sense for any ATP1A3 conditions?
Ooh, okay. I'm gonna have to look that one up because of the big expanding list of ATP1A3 conditions. I can't even say I know all of them. If this person is able to, private message you, so I can look that up and get back to them, maybe through Sharon or somebody, that would be fine. I can't directly, of course, give medical advice, but that's a simple question that I can see if there's any precedent for that. My follow-up question would be, is there any imaging or other what is called substrate, meaning basis for the facial paralysis, or is it just a mysterious facial paralysis? Because I'd imagine that most of the ATP1A3 disorders would not leave a specific mark on the brain, for example, to correlate with that facial paralysis. But that's a really great question. I'm hoping that this person has access. Sometimes it's hard for adults to see academic clinicians the way kids can. Sometimes it's hard for adults to find a neurologist who's interested in rare conditions compared to pediatric neurologists always tend to be interested in rare conditions. I was a pediatric neurologist who was so interested in rare conditions, I jumped out of pediatric neurology to do genetics, and that's less common on the adult side. So hopefully she's able to find someone who's going to take an academic and deep interest in figuring out how this might relate to ATP1A3 or not. Because that's the tricky part, is just having symptoms. If they're not ATP1A3 symptoms, it may not help build that argument for that gene change to be the cause. So I'll look that up if you can.
Okay.
Yeah, I'm asking her to email me separately,
okay, great.
so
and
we'll take care of that.
I'll check that up.
Yeah.
Great question,
if my child had genetic testing years ago, is there any reason to have updated testing today? You might have touched on this already.
So is this a person whose child got a positive result or left without a helpful diagnosis? Do we know?
We don't.
If it's positive, I would say that's not going to change. If your child has a common ATP1A3 gene change, I'd say you're done. That is, there's not really so much of a problem with false positives on genetic testing, especially if it's a gene change that's very famous, and we're not worried that we misidentified it as we called it pathogenic when it's truly a not pathogenic. But if there's not really a problem with false positives when the lab comes back and says your child has a classic ATP1A3 gene change for AHC, I think we're done. If your child got an inconclusive, the first step is actually to have your genetics provider research the inconclusive to see if in 2026 it's not an inconclusive anymore. Then you don't need checking, you just need reinterpretation. If it was negative, kind of what I said before, I think that today's lab techniques are better, and maybe they even need a broader test with other genes, like I tried to do with my one patient, except what I ended up with was getting the original gene rechecked and finding out that she did have ATP1A3 as her culprit. So I would say, yes, go back. I tell all my patients to go back to genetics every 2 to 3 years just to keep up with what's new. The geneticist thinks that is silly when you get there. You'll both know pretty fast, and you'll space out your next visit. But I think it's just really good to keep that person on your team. And I think if that's true positive, then you got a good neurologist. Maybe you don't need that. I'm sorry, to keep up with research, rather. I'm sorry, go ahead.
That's okay.
Oh,
You did talk about parents getting tested. Should siblings get tested as well?
This is a great controversial topic. So if it's de novo, then the chance of a sibling having it is probably about less than 1%. So the reason it's not zero is that it's possible for the mom or the dad to have more than one egg or sperm cell, the one that made their child who's diagnosed with AHC, and then one that could go on to make another child. Most of the time, AHC, the severe forms of ATP1A3, like AHC, has what we call high penetrance, meaning if you get the gene change, a real pathogenic gene change, you'll probably show pretty obvious symptoms. There may be some situations where the penetrance or the chance of getting affected by your gene change is less than 100%. I've got to see what the literature says for this. What I usually recommend is you're starting with that less than 1% chance that the sibling shares it, then you're decreasing it further because they don't have symptoms. I usually recommend that sibling get tested. This is not for AHC, but just in general, before childbearing to be 100% sure. Just because weird things happen, that may be a little too cautious because, again, their chances of inheriting it were low. And then if they have no symptoms, that's another lessening of their risk. But just because the stakes are high, they can maybe go more confidently into pregnancy or their partner's pregnancy by getting tested. And it's really easy to get tested. One of the labs, I won't name names, I'm not trying to play favorites, offers comparison testing for $99 self-pay. So that would be the worst case financial scenario to get that sibling tested. Again, this is being very cautious. Most likely the sibling, if the one sibling has very severe classic AHC, it's extremely unlikely that their healthy sibling is a carrier, extremely unlikely. But when we start talking about the milder ATP1A3 variants, like we're talking about a mother and daughter who might both share it, maybe it's harder to tell, even though maybe some people have childhood onset symptoms. If a couple is planning another pregnancy, then and they're the parents of a child with AHC, some people would say they look at the less than 1% recurrence risk and the next kid, and they look at that and say less than 1%. That's a better than 99% chance my next child will not have this. I'm good with that. Let's go. Other parents are going to say, I'm not good with that because it's a big deal to be a parent of a child with AHC. We love our child, but we can't imagine doing this again and having to spread our care to more than one child who has these needs. So they might choose, and again, this is a very personal decision, to test embryos. So they'll make their embryos outside the body, essentially the IVF or test tube baby. They can test the embryo and they can say, yep, this embryo is unaffected. Let's put this one into the womb and start the pregnancy. It's very expensive. It's hardly ever covered by insurance, especially when the parents aren't really carriers. So the insurance company is going to say, well, your chance is less than 1%. And then not everybody's religious or moral views says it's okay to be making and choosing embryos. So as geneticists, we would never tell people that they shouldn't just have kids naturally. If that's right for a family, that's right for a family. But if it scares people to take that less than 1% chance and they would like to expand their family with more confidence, they could use reproductive technology.
And then just really quickly back to the sibling, was that whole genome sequencing for the
no,
99?
actually, the good news is that the sibling only needs to have that exact point mutation or gene change checked. That one thing. Okay. So instead of all 20,000 genes, literally one letter needs to be checked. And that's why it's available for $99. Okay. Some labs may offer you with your original test, like this is only if the main patient has been tested recently, they'll have a follow-up program. They'll say, okay, you've got 5 months to test whomever you want. And they'll offer free testing for siblings. It's kind of a gray area testing kiddos for things. I usually say that if it's something that can affect someone in childhood, which AHC definitely can, then it's ethical to test your other kids, without asking them first and say, okay, I'm the mom, I'm the dad, I want to test these other kids, because it'll be during childhood that either the child will get symptoms or not get symptoms. If it's a condition, and there are some ATP1A3 conditions that affect adults, like rapid onset dystonia Parkinsonism, most ethicists will say, you know what, let's let that kid grow up to decide whether they want to get tested, because this could affect their life insurance, this could affect their military service, maybe they just don't want to know. And so for milder adult onset cases, we don't test healthy siblings. For severe pediatric onset things, we would offer parents to test healthy siblings most of the time. And sometimes it's very cheap. If that would put your mind at ease, I say go for it.
All right. Should families worry if they don't have one of the common ATP1A3 variants?
That's great. Great question. So I think it just may be a little longer wait to take advantage of gene therapy. And right now, nobody's getting gene therapy, to be clear. We're all on the same slope. But from what I'm reading, the most likely thing that's going to happen is that the prime editing will be tried out on the top 3 common gene changes. Again, not because these kids are more deserving or even more seriously affected than other kids, but by trying to address those first, you're going to be able to help the most people the quickest. But this technology, again, is meant to be generalizable and upscalable, meaning after that, moving on to whichever, maybe even personal private mutation, meaning nobody but you has this, and being able to access that. So I think it may be a little longer wait. It's also going to be a little harder to find statistics about your child's gene change when there's not much written about it. That can be a challenge as well. But that doesn't mean it's necessarily more severe. It just means it's more rare and unique.
You had mentioned genotype-phenotype correlations. How does that help researchers develop treatments?
That's a great question. I don't know if that helps researchers develop treatments as much as helps families and physicians and other providers know what to expect and watch for. For example, the gene change that has a lot more cardiac arrhythmias than others, you might want to put on a push to have that child more carefully followed by cardiology compared to someone whose gene change isn't associated. When they do research, though, researchers like to categorize people in groups. So perhaps they will, when they're doing a natural history study, meaning checking out symptoms, they're going to sort people out by their gene change so that they can kind of compare like things to like things. And then when it's time for gene therapy, of course, they have to try to fix each gene change one by one. So then those labels become important, and knowing what normally happens. So for example, if we're going to do a gene change for D801N, we first, an editing, try to do a gene therapy, we first need to make sure that we know what normally happens with kids and adults who have D801N. And then when we give, that's called historical control. So usually the current approach, the favorite approach, is to give the treatment to people, hopefully without a placebo, because nobody wants to be in that placebo group, that sugar pill or sham group. Give the treatment to some people who all have the same gene change, and then look back at the historical data from all those people who volunteered to give data over the years about how their child is doing. And both the people in the treatment group and the people in the historical data group have to have the same gene change, so it's apples to apples. And then they can see how much better the people, hopefully better, the people with the gene change, gene editing, are faring with the treatment. And then that group of like people, that same genotype, will be their perfect match control. And then they'll repeat that process with a different genotype, and those people with that genotype being their match control. This is the trend in current research, is to try not to give anyone placebo when you can avoid it, especially if you're dealing with a progressive condition that time is of the essence. So all the people who are in the trial hopefully will know that they're getting the active substance. And then the people who have volunteered in the past to give data will be the untreated group, but they're not actually in the current trial getting any kind of a sugar pill or sham treatment, if that makes sense. That's kind of what we're seeing the trend towards these days, which I think is much more ethical and practical, because we don't have a whole lot of people here. We only have a few people to test here, and we want to make sure as many people get the treatment. And if you're in a research study, they're going to tell you whether you're getting the active treatment or placebo. If you're possibly getting placebo, they have to tell you this. This is part of the consent process. So you don't need to fear getting a placebo against your will unless they come out and tell you that that's a possibility. That makes sense.
Out of ASO's gene replacement and prime editing, which approach seems the most promising today and why?
I think it's a kind of, my understanding, kind of a tie between ASO and prime editing. I think gene replacement is great for things like Duchenne muscular dystrophy. Those kids are missing dystrophin. We give them dystrophin, they get better. Actually, we can't give them the entire molecule because it's ginormous, but they give them a little piece of it that does the job, and then they improve. But that's not going to be a great strategy for this because we're not getting rid of the thing we want to get rid of. So I think prime editing and ASO is cheaper. ASO may be a little further along. It's an older technology. Making those little fragments, that's actually very cheap. There's a lot of tech that goes into it, and giving it to humans and making them human grade is, I guess, a little challenging. But the actual production of the fragments is a technology that's been worked on for a while for other applications, and it's cheap. The prime editing is really cutting edge. So I'm imagining that's going to be a little more expensive and a little harder to get started. I love the idea of prime editing because it's permanent, and ASO is less permanent. So I think they both have promise. What we're seeing right now in spinal muscular atrophy, which is a little crazy, the gene therapy, which is very expensive, and the ASO, which is also very expensive, some families have been able to get their insurance company to give the child both. So the child both gets their gene fixed, and also, because it's not 100% of a fix, they also take the ASO therapy. They do both. And that's really powerful. And SMA has been a huge success story. SMA, I'm off topic here, but that was a fatal disease until 2016. And in 2016, they got the ASO. In 2016, they decided to put it on the newborn screen so we could catch kids early and get them the ASO. By 2019, we had the gene therapy. And the Cure SMA is the parent group. They are not letting off. I'm kind of jealous because, like, wow, SMA has this and SMA has that, but they're still going. They're still trying to come up with something better at every turn, even though they have more stuff than all the rest of us, and I'm jealous. So I would love us to be like them. And that's a wonderful problem to have. But I mean, I'm old enough to remember families making terrible decisions about their child, ventilator versus hospice, essentially. And now it's like, here's your gene therapy. And the crazy part is it's $4 million, but the insurance companies are paying for it. That's like, wow. Because they realize that the lifetime care of a child with a serious neuromuscular condition, because it's a muscle disease that affects breathing, is expensive. These kids are in the ICU a lot with their pneumonia and things like that. So I hate to say that economics should be the driver of whether or not a child gets a life-saving therapy, but insurance companies are driven by economics, and yeah, they're paying for this stuff. So in a clinical trial, your treatment is free, but once it gets approved by the FDA and your doctor's prescribing it, it's not free anymore.
So with the prime editing, we're hearing a lot about that.
Yeah.
What do you think families should realistically expect over the next several years?
I can't tell. I wish I could tell. I'm going to say all my knowledge of prime editing goes back exactly 1 week now to the 21st when I was watching television and my mother texted me this New York Times article. And typically she texts me articles that are, about cats or something, or that I've already read. And then here's this article, I'm like, oh my gosh, it's the Frost family. And oh my gosh, it's prime editing and it's coming. And I'm giving a talk next week and I'd better update those slides. And then that launched me into reading about it, to talking to Al George about it. So yeah, I'm not on the front lines of it. I've read what you've read with a little bit of insider because I have access to Dr. George, but I'm really amazed that you can go to their website. I put the name as Center for Therapeutic Genetics. You can go to that Center for Therapeutic Genetics website and see what they're doing. But it's not ready yet. And the article is my source of the information that they're planning, at least the AHC community, because it featured AHC. That's what the article is about. It's about a couple other conditions like Dravet, which is a very serious epilepsy disorder. But AHC, I was super excited to see us featured and to see Annabelle featured and in her beautiful pictures. But it did imply that they're going to start with the common gene changes. And that's in the medical field, that's usually considered a responsible thing to do because we really want to help as many kids as possible. But we don't want to leave out those who are unique. I'm a big believer in personalized medicine. This is the ultimate personalized medicine, right? Or down to the DNA level for kids who are unique in their mutations. So I think we'll get there, but I cannot put a timeline on it. I'm just amazed that it's come up so fast in the last year. I'm sure it didn't just pop up. I'm sure that they've been working on it for quite a long time, but the fact that it's now in the public eye is impressive. And I think that we've got some momentum going.
Okay. That'll be it for questions, unless anybody has any they want to add to the chat. You do have a couple of, thank you so much for that explanation. Thank you, Dr. DeBrosse, for your excellent presentation and clarifications.
She's the best.
Oh, thank you.
So I will echo those comments. Dr. DeBrosse, thank you so much for being with us tonight and for sharing your expertise with our community. I know how much work goes into these presentations, and you've always been such a great partner for us. So thank you for your dedication to the AHC community and all of the patients and families that deal with AHC. You are a valued partner to us. So thank you. I'd also like to thank all of the people that have joined tonight. Thank you for your incredible questions. What a great Q&A session that was.
Great.
It was very robust and gave so much information. So thank you. And for those of you who are watching this as a recording, especially for our global community, because this is a very difficult time. It's difficult for everyone with an HC child, let's be honest. We thank you for engaging in the community, and we hope that you get so much from this webinar. As an AHC mom, personally, I just know firsthand how overwhelming genetics can be. And so that's why we value Dr. DeBrosse so much, because she has such a great style of kind of breaking it down and making it understandable. I think my hope for parents who are watching this, or caregivers or medical professionals, is that, well, not medical professionals because they know, but for parents and caregivers that,
I tell you the
yeah, that
it's
a little less intimidating, and that you have, knowledge is power, and that you feel less intimidated and more confident advocating for your AHC child or AHC adult, and having those conversations with your medical care team. And that's why we put on these educational series and these webinars that we do, is really for our community. So I hope that you get so much out of this webinar. If you would like to watch this webinar, as with all of our other webinars in the educational series, it will be on our website within 1 or 2 days, typically. Our website, for those who don't know, www.ahckids.org. It will also be posted on our socials, Instagram and Facebook, and we'll also be, we'll send it out via email as well. Last, if you don't follow the HC Foundation on our socials, yes, we have a robust website. We communicate via email, but I would say the most active form of communication that we use is our social media channels. So if you are not following us yet on Facebook or Instagram, please do so, because we share so much information for the HC community. Beyond webinars, we have research updates, we have community resources, family resources, foundation news, and upcoming events, one of which is our signature event, which is our AHC family meeting. Was a very proud moment for us. We created it 30 years ago. We have it every 2 years, and we had a great time in Cincinnati in 2025, and we're planning our next one in Houston for 2027. And we're already sending out updates and news links and all sorts of things for Houston. So we hope you join us in Houston, but please follow us on our social media channels to stay updated. And that's a wrap on this webinar. Thank you again for being here. Have a wonderful evening, and we look forward to seeing you at our next webinar.
Thanks for having me.
Thank you,
Thank
Dr.
you. Great
DeBrosse.
questions.
Good night.
Okay.
Good night.
Bye.
Take care.